Hi, my name is Jiaying and I am a graduate student at Brown University. Today I would like to introduce a method called LYRUS to predict pathogenicity of missense variants. Differentiating pathogenic single amino acid variants, or SAVs, from neutral ones is of great importance as it can enhance our understanding of the correlation between genotype and phenotype, facilitating the development of novel treatment strategies for complex diseases. Our model incorporates the essential properties of evolutionary conservation, structural information, and protein dynamics to predict pathogenicity of SAVs. In this study, we were able to determine the best optimal machine learning model tailored to our dataset for predicting the pathogenicity of missense variants with high accuracy, but also uncover some of the underlying structural properties associated with pathogenic proteins. CleanVar data was used to generate our training dataset. We selected missense variants with a review stars of at least 1. The total number of SAVs whose feature values were all obtainable is 20,307. Variation number is a recently developed sequence feature. It is a phylogenetic measure that can quantify sequence conservation using sequence homologs from different species. The figure here displays the pipeline for calculating variation number. A smaller variation number suggests more conservation, so if a substitution takes place at this position, it is more likely to be pathogenic. The figure here shows the histogram of variation numbers for the 20,307 pathogenic and neutral SAVs. Variation numbers range from 0 to 1, with 0 being the highest conservation value. The t-test results suggest that pathogenic SAVs are more conserved than the neutral ones. A total of 15 features, which includes 5 sequence-based features, 6 structure-based features, and 4 dynamics-based features, were used as our feature set. We calculate the Pearson's correlation of all possible pairwise combinations of the 15 features. Only 4 pairs had correlation coefficient greater than 0.5, suggesting that the selected features were largely independent. We use TPA, which stands for Tree-Based Pipeline Optimization Tool, to pick the best performing machine learning pipeline. The best machine learning model suggested by TPA is an XGBoost classifier, which is a scalable and accurate implementation of gradient boosting machines. Principal component analysis was applied to our feature dataset with the objective of cross-validating our feature selection and checking redundancy among our 15 features. The plot here shows that at least 13 components are needed to describe 90% of variance in the calculated results of the 15 features. This analysis validates that there is minimal redundancy in our dataset and further supports the use of the selected features in our machine learning model. Feature importance analysis suggested that sequence-based features had higher importance score than structural and dynamic-based features. We calculated the accuracy, sensitivity, specificity, F1 measure, and MCC for LYRUS as well as 9 other predicting methods. Our model achieved the highest accuracy, specificity, F1 measure, and MCC. The sensitivity of LYRUS was slightly lower than that of MVP, Polyphen2, and SIFT. To further test the ability of different methods of distinguishing pathogenic mutations, we plotted both the ROC and the PR curve. LYRUS has the highest AUC score of 0.941. It also dominates the PR space with a score of 0.95. Subsequently, we did a case study on P10 protein, which is associated with metastatic cancer. 133 SAVs were used to test the performance of LYRUS against other software. From the bar chart, LYRUS achieved comparable performance as other software. We conducted an additional case study by applying LYRUS to TP53 protein. Based on our prediction displayed in this heat map, the region spanning position 100 and 290 is predicted to be highly pathogenic. This region contains the core domain of the TP53 protein, and mutations in the core domain can result in the loss of DNA binding activity. Additionally, more than 80% of somatic TP53 mutations in human cancer occur in this region. These findings validate our prediction. Our study introduced LYRUS for predicting the pathogenicity of SAVs. The most predictive features are sequence-based features. The high impact score of variation number also suggests the effectiveness of this novel feature for categorizing pathogenic and non-pathogenic SAVs. Among the structural and dynamic space features, changing folding free energies and the location of binding sites also shows strong contribution to the high predictive power of our model. Finally, one area for future improvement is the incorporation of structural changes caused by mutations into our model. It has been proven that mutations promoting protein misfolding contribute to a variety of human diseases. Incorporating information related to structural change, such as RMSD, may facilitate pathogenicity prediction. That's essentially the summary, and thank you for coming to my presentation.